In this video, we are going to discuss a tutorial question which is on chapter Cellular Respiration, part B, structured question. For question number 1, it gives you a figure which is figure 3 that shows the glycolysis process in a eukaryotic cell. So as you know, glycolysis is the first stage of cellular respiration and it occurs in the cytoplasm. And it has 10 reactions. So for glycolysis, it has two phases, which is the first one. The first phase is from glucose until it produces two molecules of X. So what is X? I will explain later. So the first phase is energy investment phase. Okay, so this phase involves reaction 1 until reaction 5. Okay, so for the second phase, from molecule X until it produces two molecules of pyruvate, which is energy payoff phase. Okay, so this involves reaction 6 until reaction 10. So why do we call it as energy investment phase? So in this phase, two molecules of ATP is used. Okay, so why do we call it as energy payoff phase? Because it produces 4 molecules of ATP. So for question A, identify which part of cell this process occurs. Obviously in the cytoplasm. So if you look at in this figure, because glycolysis involves the usage of two molecules of ATP and produced 4 molecules of ATP, you have to know what is the net production of ATP for glycolysis. So net production of ATP molecule is 4 ATP produced during payoff phase minus 2 that is used during investment phase. So the net production of ATP will be 2 molecule of ATP, okay, for glycolysis. Alright. So for the next question, list the net products of glycolysis. So the net product of glycolysis will be 1, 2 molecule of ATP, from the 4 that you minus with 2, and then 2 molecule of NADH. So NADH molecule that is produced during glycolysis is a very important molecule that will be used during the fourth stage of cellular respiration, which is oxidative phosphorylation. Oxidative phosphorylation will produce more ATP molecule for each NADH that enters into the oxidative phosphorylation. So for one molecule of NADH, during oxidative phosphorylation, it produces 2.5 molecule of ATP. Okay, so another product of glycolysis is 2 molecule of pyruvate. 2 molecule of pyruvate. So question C, name molecule X and explain how molecule X are produced. So molecule X, if you look at the question, the mark for this question is 3 marks. So for the first point is you have to state what is X. So X is G3P. Okay, so for the next 2 marks, you have to explain how the X molecule, the 2 X molecule are produced. Okay, so if you look at in this diagram, so you have this molecule, fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate is a 6 carbon sugar. Okay, and it has 2 phosphate groups attached to it. Because it has 2 phosphate group attached to it, it is a highly energized molecule and it has to be broken down into 2 molecules of 3 carbon sugar. So molecule X, which is G3P, is a 3 carbon sugar. Okay, is a 3-carbon sugar. So in order to break fructose 1,6-base phosphate, it requires an enzyme. We call it as aldolase. Okay, so aldolase will break fructose 1,6-biphosphate into 2 molecules of 3-carbon sugars, which is one is G3P and the other one is DHAP. DHAP stands for dihydroxyacetone phosphate. Okay, dihydroxyacetone phosphate. So to proceed into the next phase, which is phase 2, DHAP has to be converted into its isomer, which is G3P. So now you have 2 molecules of G3P. Okay, so it means that for you to gain the next 2 marks, okay, you have to mention, this is for the first mark, okay, this is for the second mark. Fructose 1,6-base phosphate, 1,6-base phosphate has to be broken down, okay, using the enzyme aldolase, okay, into 2 molecules of 3-carbon sugars, okay, which is one is G3P and the other one is DHAP. Okay, so that is for the first mark. You have to mention the enzyme aldolase, and then the product here, which is G3P and DHAP. Okay, so first mark, second mark, okay, third mark. So you gain 3 marks. So you can also mention the in order to get 2 molecules of G3P, the DHAP will be converted into G3P by the enzyme isomerase. Okay, isomerase convert DHAP into G3P, then you will get 2 molecules of G3P. Okay, which is the X molecule. Okay, question D. In the given reaction, identify which molecule is the oxidizing agent and which is the reducing agent. So in the glycolysis process, you know that X is a molecule, we call it as G3P, okay, glyceraldehyde triphosphate. So and then you see this NADH. NADH is formed from this molecule, which is NAD plus. Okay, so in order NAD plus to form NADH, it means that this molecule has to be reduced. Okay, so NAD plus will accept hydrogen ion and also electrons. So it means that it is reduced to become NADH. So if NAD plus is reduced, it means that it is an oxidizing agent. Okay, so if it's an oxidizing agent, it means that it has to reduce the molecule before it, so which is G3P. Okay, so it means that G3P will be oxidized. Okay, oxidized. So because G3P is oxidized, it means that G3P is a reducing agent. Okay, reducing agent. G3P is oxidized because it loses its electrons to NAD plus molecule, so that NAD plus becomes NADH. So G3P is a reducing agent, NAD plus is the oxidizing agent. Question E. What happens when ATP is dephosphorylated? So you have to understand what is dephosphorylated. Dephosphorylated, it means that the substrate or the molecule loses a phosphate group. Okay, loses a phosphate group. So in this case, the molecule is ATP. ATP stands for adenosine triphosphate. The molecule has 3 phosphates. So the ATP molecule will lose its third phosphate. Okay, the third phosphate is removed through the process of hydrolysis, and then the process that involves the removal of phosphate group releases energy, and the process is exergonic reaction, and then it produces ADP molecule. Okay, so that is what happened during dephosphorylation. So the question is 2 marks. Okay, so for the first point, ATP removes a phosphate group. Okay, through the process of hydrolysis, the reaction releases energy, and then the process is hexagonic, and then also produces it produces ADP molecule. Okay, ATP becomes ADP. Okay, ADP is produced. Question F. Name the process that generates ATP in glycolysis. So the production of ATP during glycolysis is through substrate level phosphorylation. Okay, so it means that to produce ATP molecule, originally it is from the molecule ATP. Okay, so to become ATP, it receives a phosphate group from a substrate. So that's why it is called as substrate level phosphorylation. But the production of ATP through substrate level phosphorylation only produces a small number of ATP molecules. So as you can see, for glycolysis, the production of ATP through substrate level phosphorylation only produced a net of 2 ATP molecules. Another process that also produces ATP through substrate level phosphorylation is citric acid cycle. Okay, so for a complete cycle of citric acid cycle, it only produces 1 molecule of ATP. Okay, so but in order for cellular respiration to produce a high quantity of ATP, the production of ATP that produces a high number of ATP is during oxidative phosphorylation, which is the 4th stage of cellular respiration. So cellular respiration will produce more ATP during oxidative phosphorylation compared to substrate level phosphorylation. So this is the same question. Okay, figure 4 below shows pyruvate oxidation. So pyruvate oxidation is the 2nd stage of cellular respiration. So in this stage, it involves pyruvate, which is a 3-carbon sugar, to be converted into acetyl-CoA, which is a 2-carbon sugar, and it occurs in the matrix of mitochondria. Okay, so in this reaction, it also involves the removal of carboxyl group of pyruvate and becomes carbon dioxide. Okay, and in the process also occurs the production of NADH molecule from NAD plus molecule. Okay, so basically so this is what happened during pyruvate oxidation. So why is the above step is important before it enters into the Krebs cycle? Krebs cycle is the 3rd stage of cellular respiration. So why is it important? Because the reaction will convert pyruvate, okay, into acetyl-CoA, and once acetyl-CoA is produced, then only it can enter into the Krebs cycle to combine with oxaloacetate, which is a 4-carbon compound, and then convert it into citric, okay, which is a 6-carbon compound. So it is important because to convert pyruvate into acetyl-CoA, then only it can enter into the Krebs cycle to combine with oxaloacetate to form citrate. So question C. Explain how pyruvate molecule is oxidized in the above reaction. So oxidized, it means that the pyruvate molecule will release its electron and gives the electron to NAD plus, and NAD plus will become NADH. So if you look at the question, it is 3 marks. Okay, so first you have to mention that pyruvate, as I've said before, pyruvate will undergo, okay, it will undergo decarboxylation. Okay, what does it mean? The carboxyl group is removed as carbon dioxide. So that is the first point. Okay, carboxyl group of pyruvate is removed as carbon dioxide, which is decarboxylation. So that is the first point. For the second point is you mentioned this one. Okay, carbon dioxide is removed or is released. And then for the third point, the remaining two carbon molecule, which is now is oxidized, it will form acetate. Okay, acetate is formed because the molecule now is oxidized. And then for the third point, the electron are transferred to NAD plus and becomes NADH molecule. So the next things that happen is that a coenzyme A will be added to the acetate molecule, forming acetyl-CoA. Okay, so you have several points. The question only wants 3 points for 3 marks. Okay, question D. Figure 5 below shows Krebs cycle which occurs in the mitochondrial matrix. Krebs cycle is also known as citric acid cycle, and it is the third stage of cellular respiration, and it occurs in the matrix of mitochondria. So in the Krebs cycle, the acetyl-CoA that was produced during the link reaction or pyruvate oxidation stage will release its CoA, and then the remaining two carbon molecule will combine with oxaloacetate to form citrate. Citrate will be converted into its isomer, which is isocitrate, and then isocitrate will be oxidized to form alpha ketoglutarate. Okay, so isocitrate that is oxidized will release its electron and received by NAD plus, and NAD plus will be reduced to form NADH. Okay, so isocitrate and citrate is a 6 carbon compound and alpha ketoglutarate is a 5 carbon compound. So it means that during the conversion of isocitrate to alpha ketoglutarate, the carbon dioxide, one molecule of carbon dioxide will be released. And then for alpha ketoglutarate, it also will be oxidized. It means carbon dioxide will also be removed during this stage, and alpha ketoglutarate will be converted into succinyl-CoA. So succinyl-CoA is a 4 carbon compound. So during the oxidation of alpha ketoglutarate into succinyl-CoA, one molecule of NADH will be produced. Okay, so succinyl-CoA will be converted into molecule E, which is succinate. Okay, succinate, and during the conversion, one molecule of ATP will be produced. So it means that during this reaction, one inorganic phosphate will be added to succinate, and then the inorganic phosphate will be given or received by GDP. GDP becomes G3P. Okay, guanosine triphosphate, and then the phosphate of GTP will be given to ADP, and ADP will become ATP. Okay, succinate, which is molecule E, will be converted into fumarate. Okay, so fumarate is a 4 carbon compound. Succinate is also a 4 carbon compound, and during the conversion of succinate to fumarate, okay, fumarate, one molecule of FADH2 is produced, and then fumarate will be converted into malate, and then malate will be oxidized to form oxaloacetate. So during the conversion of malate to oxaloacetate, one molecule of NADH is produced. Okay, so for question D, okay, name molecule A, D, C, and E. So we have named all the molecule actually. Okay, so A is acetyl-CoA, B is citrate, C is alpha ketoglutarate, F is fumarate. Okay, so the question is 2 marks. So it means that for every 2 answer with the correct answers, okay, you get 1 mark. Okay, 2 correct answer for 1 mark. Okay. Okay, so question E, when molecule D is changed to molecule E, ATP is produced. Okay, so D is succinyl-CoA, E is succinate. Okay, so during the conversion, 1 molecule of ATP is produced. So the question is 2 marks. So during this stage or this process, CoA in molecule D or CoA in succinyl-CoA will be displaced by inorganic phosphates. Okay, succinyl-CoA will have a CoA attached to it. This CoA will be removed and replaced by inorganic phosphate. That is for 1 mark. Okay, for the second mark, phosphate group is then transferred to GDP and to form GTP. Okay, so that is the second mark. The third mark, you can write the phosphate of GTP will be given to ADP to form ATP. Okay, so those are the marks that you can include in your answers. Question D2. ATP production requires energy. Okay, requires energy. State where this energy is obtained from. Okay, so in order to form ATP, okay, from inorganic phosphate and also ADP, a form of energy has to be inserted into the reaction to make bond between inorganic phosphate and the molecule ADP. So where is this energy come from? So the energy comes from the hydrolysis of GTP molecule. Okay, hydrolysis of GTP molecule, which is hexagonic, which releases energy. Okay, which releases energy to make bond between the inorganic phosphate and also ADP to form ATP molecule. List all the products of Krebs cycle obtained from one glucose molecule. Okay, so the key word here is one glucose molecule. So you remember, for one cycle of Krebs cycle, okay, the product is 3 NADH, 1 FADH2 molecule and 1 ATP molecule. Okay, 3 NADH, 1, 2 and 3. 1 FADH here and 1 ATP here. Okay, but the question asks for 1 molecule of glucose. So it means that the Krebs cycle has to occur 2 times. Okay, because 1 molecule of glucose during glycolysis, it breaks into 2 pyruvate. Into 2 pyruvate and then pyruvate into acetyl-CoA, and each acetyl-CoA enters into 1 cycle of Krebs cycle, 1 cycle of Krebs cycle. Okay, so for 1 glucose, the Krebs cycle has to occur 2 times. So you have to times all this product by 2. Okay, so the answer will be 6 NADH, okay, 2 FADH2 molecule and 2 molecule of ATP. Question 3. Describe 2 differences between glycolysis and Krebs cycle. Okay, so glycolysis, it occurs where? In the cytoplasm. Okay, Krebs cycle, it occurs where? In the matrix of mitochondria. Krebs cycle, that is for the first mark. Okay, for the second mark, another difference between Krebs cycle and glycolysis is it is for glycolysis, it is a linear process. Okay, it is a linear process because it involves the reaction that convert glucose into 2 molecule of pyruvate. Okay, so it is a linear process. But as for Krebs cycle, it is a cyclic process. Okay, cyclic process because acetyl CoA will enter into the cycle, combine with oxaloacetate, and oxaloacetate will be regenerated, will be reformed again. Okay, will be reformed again to start the cycle again. Okay, so that is Krebs cycle. Krebs cycle is a cyclic process. Glycolysis is a linear process. Another differences that you can add into your answer is by giving the net product of glycolysis and also the product of Krebs cycle. So the product of glycolysis is 2 ATP, a net of 2 ATP, and then it also produces 2 NADH molecule and also 2 pyruvate molecule. But as for Krebs cycle, it produces for 1 glucose. Okay, is the Krebs cycle produces 6 NADH. Okay, and then 2 ATP molecule and also 2 FADH2 molecule. The process also occurs and produces 4 molecule of carbon dioxide. Okay, so these are the product of the two processes, glycolysis and also Krebs cycle, for 1 molecule of glucose. Okay. Figure 6 below shows electron transport chain in a mitochondria. State where the protein involved in the electron transport chain are embedded. Okay, obviously it is embedded in the inner membrane of mitochondria, or another term of it is cristae. Okay, so that is for 1 mark. Question C. NADH and FADH2 shuttle high energy electrons from glycolysis and Krebs cycle. Write the pathway of electrons for both carriers along the electron transport chain, NADH and FADH2, as you see here. Okay, so these are actually electron carriers. Okay, they carry high energy electrons. For NADH molecule, so the NADH molecule will release its energy, its highly energized electrons, okay, to the components of the electron transport chains that are embedded in the cristae. So the electron carriers that are embedded in the cristae are complex 1, complex 2, complex 3, complex 4. And then you have P and Q, they are mobile carrier. P is ubiquinone, Q is cytochrome. For NADH molecule, okay, the pathway of the electron will be the electron of NADH will be released and received by complex 1. Complex 1 will pass the electrons to P, okay, or ubiquinone. Okay, ubiquinone. And then ubiquinone will pass to complex 3, and then will pass to Q or cytochrome, and then pass to complex 4, and then the last electron acceptor will be oxygen. As for FADH2, okay, FADH2, the electrons of FADH2 will be received by complex 2, okay, complex 2, and then pass to ubiquinone or P, and then pass to complex 3, and then to Q or cytochrome, and then to complex 4, and then the last electron acceptor will be oxygen. Okay, oxygen will receive the electrons and combine with hydrogen ion, and then it forms water molecule. Question D. As electrons are transferred along the electron transport chain, energy becomes available. Explain how this energy is used in ATP production. So for this process, you have to explain on chemiosmosis. Okay, chemiosmosis. So in order for you to explain chemiosmosis, you have to know the region within the different region within the mitochondria. So this region here, okay, is the matrix of mitochondria, and it has a low concentration of hydrogen ion. This side here is the intermembrane space. Okay, that has a high concentration of hydrogen ion. Complex 1, complex 2, complex 3, and 4 are electron carriers, and they will also function as proton pump. Okay, this one here you see is ATP synthase. Okay, so these are the components of the electron transport chain that you have to know in order to help you to explain on chemiosmosis. Okay, so the question is worth 3 marks. So basically the question wants you to explain on the process of chemiosmosis, how energy from the electron is used to produce ATP molecule. So for the first point, you have to explain that the transfer of electron from one carrier to another carrier releases energy. Okay, so this energy is exergonic. Okay, it's exergonic. It releases energy, and the energy is used to pump hydrogen ion using the proton pump. Okay, to pump hydrogen ion from the matrix into the intermembrane space. Okay, the energy from the electron is used to pump hydrogen ion from the matrix into the intermembrane space because it is from low concentration to high concentration region. Okay, so that is for the first point. Okay, energy from the electron is used to pump proton using proton pump. For the second point is you mentioned from where to where? From matrix into the intermembrane space. So that is for the second point. For the third point, uh, uh, is there you mentioned there is a high concentration of hydrogen ion in the intermembrane space that will create, okay, proton motive force. Okay, that will create proton motive force, and then this force will drive back the hydrogen ion back into the matrix through the ATP synthase. So ATP synthase will function as protein channel, okay, channel that allows hydrogen ion to diffuse, okay, diffuse back into the matrix. Okay, so as hydrogen ion diffuse back into the matrix using ATP synthase, okay, ATP synthase will use the exergonic flow of hydrogen ion to phosphorylate ADP, okay, to form ATP. ADP combines with inorganic phosphate to form ATP. So here I've listed 4 points. Okay, you just choose any 3 points for 3 marks. Question E. Explain the role of oxygen in the electron transport chain. So oxygen will function as what? As the last electron acceptor or the final electron acceptor. So as the electron travels from one carrier to another carrier to another carrier to another carrier, the electron will be received by the last electron acceptor, which is oxygen, and then the oxygen will combine with hydrogen ion to form water. Okay, so oxygen is the most electronegative molecule. So that is for the 2 mark. The first mark, oxygen as the final electron acceptor. Second marks is you mentioned it is the most electronegative molecule compared to the other electron carriers of the electron transport chain. Question number 4. The number of ATP produced are different depending on the type of shuttle used to transport electrons from cytosol into mitochondrion. Okay, so you have to know that during glycolysis, it produces two molecules of NADH, but it is produced in the cytoplasm. Okay, in order to enter into the mitochondria, NADH will just pass its electrons to electron shuttle, and then the shuttle, and then the electron will either be passed to NAD plus molecule or FAD molecule. NAD plus if it's the liver cell, okay, FAD if it's the brain cell. So NAD plus will become NADH. FAD will become FADH2. So the total number of ATP produced for liver and brain cell will be different. For NAD molecule, so for liver cell, the number of ATP produced is 32 ATP, and as for liver, the number of ATP molecule is 30 molecules of ATP produced at the end of cellular respiration. So using this table, you have to calculate, show your calculation, how do you get the different number of ATP produced for brain and also liver cell. Okay, so as you know, glycolysis is the first stage of cellular respiration that occurs in the cytoplasm. Okay, for brain cell, the two NADH produced will be converted into two molecule of FADH2 molecule. Liver cell will still maintain two molecule of NADH molecule. Okay, so you have to remember for one molecule of FADH2, it produces 1.5 molecule of ATP, and for one molecule of NADH, it produces 2.5 ATP molecule. Okay, so you have to times, okay, the 2 NADH2 by 1.5. So this will yield 3 molecule of ATP. For liver, the 2 NADH you will have to times by 2.5. So this gives you 5 molecule of ATP. Okay, so the 2 ATP molecule here is the production of ATP through substrate level phosphorylation during glycolysis. Okay, so this is 2. And then for pyruvate oxidation, which is the second stage of cellular respiration, it produces the same for both brain and liver, which is 2 molecule of NADH. So you have to times by 2.5, you will get 5 molecule of ATP. So the third, okay, stage of cellular respiration is citric acid cycle, okay, which occurs in the matrix of mitochondria. So for citric acid cycle, you have to times it by 2, because the whole calculation you have to count for the production of ATP for one molecule of glucose. Okay, so citric acid cycle has to occur 2 times. Pyruvate also has to occur 2 times. Okay, so for 2 times citric acid cycle, it produces 6 molecule of NADH that you have to time by 2.5. You will get 15 molecule of ATP. Okay, so FADH2 you have to times by 1.5. You will get 3. And then this is the last one here is this production of ATP through substrate level phosphorylation during citric acid cycle, which is 2. Okay, so now we want to calculate the number of ATP for brain cell. Okay, brain. So I give you here the color green, okay, to calculate the number of ATP. So brain 3 ATP, okay, plus 2 ATP through substrate level phosphorylation. Remember, do not add this ATP, because this one, the 5 molecule of ATP is for liver. This one is for liver. Okay, this one is for brain. Okay, so the 2 molecule of ATP through substrate level phosphorylation is this one, and then you add 5, and then you add 15, and then you add 3, and then you add 2. Okay, so the total number should give you 30 molecules of ATP. For liver cell, okay, which is the purple color here, okay, liver. Okay, you start adding the number of ATP for this one, okay, ATP. So 5 molecule of ATP, add 2, okay, during substrate level phosphorylation, add 5, okay, during pyruvate oxidation, okay, from NADH from pyruvate oxidation, and then 15, okay, and then 3, this one here, and then add by 2. Okay, so this should give you 32 molecules of ATP. Okay, so that's the amount of ATP produced for liver and brain cell during cellular respiration. So that's the end of discussion for chapter 5. I hope that you understand all the questions that we have gone through. Thank you.